Welcome to this screencast where I'll be discussing the experimental evidence supporting the semi-conservative model of replication for the DNA molecule. So you should be able to very clearly describe the structure of the DNA molecule, understanding the anti-parallel nature of the molecule, as well as the basic structure of the two sides of the molecule. By the end of this screencast, hopefully you'll be able to describe and explain the semi-conservative model of the DNA replication, and more importantly, what is the experimental evidence from Messelson and Stahl that supports the semi-conservative model of the DNA replication. So in a previous screencast, we had talked about how Watson and Crick's paper had suggested that the structure of the DNA molecule immediately suggested to them a copying mechanism. Now, we know that the copying mechanism is a semi-conservative model here, but to consider the other two models, you have this conservative model where the original parent DNA molecule could separate, the hydrogen bonds could separate, and then each side used as a template, but then the new ones would come back together. So you'd get new, and you'd get original and original coming back together again. And then this would happen subsequently, right? So original, and new, and then this would be like new sub 2, or actually new sub 1, new sub 1, new sub 2, new sub 2, new sub 2, new sub 2, etc., right? And so on and so forth. Sorry about that. And the third model might have been the dispersive model, where you had the original DNA molecule that would be sort of just split up and used as a template, but then you'd have a mixture of old and new strands here, and that would keep happening, old and new, on down the line. So to Watson and Crick and other researchers, they didn't know, was it this one or this one or this one? Was it conservative? Pardon me, semi-conservative or dispersive? We know that it's a semi-conservative model, so the classic experiment that gives us evidence to say that to support the semi-conservative model is done by Messelson and Stahl. So the semi-conservative model is the mechanism for replication. So a little bit of background here and then their experimental setup. They take bacteria over here that are cultured in a medium containing heavy nitrogen. Now, this nitrogen 15, nitrogen is normally, this is an isotope. And so this means that it has one extra neutron. Right? So it is heavier. Right? It's still nitrogen, right? But what's the big deal with this? Well, the nitrogen 15 gets into the new nucleotides made by cells. Right? So if they culture these bacteria, right, in culture means to grow, right? They grow them up in this bacteria, and several generations of growth in there, that all the bacteria in there are going to now have heavy nitrogen incorporated into their nucleotides, into the nitrogen base of the nucleotides. So they are now essentially tagged with this. So they're able to their DNA is a little bit heavier than DNA that is made with nitrogen of a lesser isotope, or a lighter, I should say, isotope. So they take these bacteria that have been cultured in this heavy nitrogen. They then transfer them into a medium that contains light nitrogen. Right? Immediately, they put them in here. And so this medium is a growth medium, right? And this medium is these cells are using to grow. Right? It has sugars and proteins, and then also a source of nitrogen for these bacteria to make their own new nucleotides. So they do this. And they know that these bacteria replicate, right, in ideal conditions, so that is like at 37 degrees C. Like every 20 minutes they're going to replicate. So they transfer those bacteria in, wait 20 minutes, and then take a sample out. Right? So after this, right, first round of replication, pardon me, they then centrifuge. And again, centrifuging the heavier ones to the bottom in the pellet, right? And then the lighter things go to the top. So after centrifuging, after one round of replication, this is important that these are the results of the first round of replication at the top here. they see that so these are all predictions here, right? So these are hypotheses, right? So if the model is conservative, then they should see this. They should see a light band and a heavy band. Right? But turns out they do not see this. Right? They see an intermediate band. Right? So if it's semi-conservative, right, then this is what they would see. This should be, then. So if conservative, then this. If semi-conservative, then this. If it was dispersive, they would also expect to see this middle band. And this is indeed what they see. They see a band of intermediate weight here. But now, in order to determine the difference between it being semi-conservative and dispersive, they have to do a second round of replication. So they wait another 20 minutes, and they take a sample from here. Right? So this is after 40 minutes, the second round of replication. And they know that it's not semi-conservative. They know that it's not conservative, right? So if it was conservative, then they would have expected to see this, right? And they didn't see that. If it was semi-conservative, then they would expect to see lighter bands, and then not so much intermediate bands, but or intermediate bands here as well. And this is indeed what they see. And if it was dispersive, then they would expect to see these results here, but they do not see that. So this confirms for them that it is the semi-conservative model of replication. Now, to see if you understand this, can you predict down here, right, if they let this go for another 20 minutes or another like 60 minutes, right? So what would you expect if it was conservative? Right? What would you expect to see if you centrifuged it? If it's semi-conservative, right, what would you expect to see? And if it was dispersive, what would you expect to see? Right? So this is a way for you to test yourself and know, right, see if you understand actually what was happening here. Okay? So do that, bring that to class, watch these animations that will support what I've discussed here, and yeah, we can talk about that in class. Okay, so you should now be able to describe the semi-conservative model of DNA replication and how it is different than the conservative versus the dispersive. So what is the difference between those? And then what is the experimental evidence from Messelson and Stahl that supports the semi-conservative model, right, of DNA replication? Okay, thanks for listening. Bring your questions to class.